Consider maybe the darkest paradox in modern neurology.
Oh, wow. Starting heavy today.
Well, I mean, it is. Because on one hand, you have high-grade pontine gliomas. These are pediatric brain tumors that are just overwhelmingly lethal. They aggressively hijack the nervous system.
Yeah, they're devastating.
But then on the flip side of that exact same system, you have the human spinal cord, an area that is under constant mechanical stress, but it possesses this almost miraculous ability to rapidly remyelinate and heal itself after minor traumas.
Which is incredible when you think about it.
Exactly. And for decades, these two things, this fatal tumor and this superpower healing, seemed entirely unrelated.
Completely different fields of study, usually.
Right. But what if the very epigenetic architecture that gives your spinal cord that evolutionary superpower to heal, what if that is the exact same backdoor that this pediatric cancer uses to tear it down?
It's a terrifying thought.
It really is. Welcome to another deep dive. Today, we are pulling apart a truly monumental 2026 paper from Nature Neuroscience by Cobby and colleagues.
Such a massive study.
And it is going to, fundamentally change how you view the biological software running in your own nervous system.
It completely reframes our entire understanding of cellular maturity, honestly.
Right.
Yeah, Cobby's team, they didn't just map the adult human central nervous system. They dug into you could call them regulatory ghosts haunting our glial cells.
I love that phrasing. Regulatory ghosts.
It fits perfectly. We're talking about mapping the chromatin state of over 100,000 individual nuclei across 20 human donors.
100,000.
Yeah. And the central revelation here is that mature cells, specifically oligodendrocyte precursor cells, which we call OPCs, and mature oligodendrocytes, or MOLs, they maintain this decoupled embryonic epigenetic memory.
Like they literally hold on to their past.
Physically hold on to it. Their developmental past is just sitting there in the genome.
Which totally implies our genome isn't just some static blueprint, right? It's a historically informed landscape.
Exactly. It remembers.
Okay, let's unpack this alphabet soup, because to understand this cellular memory, we have to look at how they bypass the traditional limitations of single-cell sequencing.
Right, because working with human postmortem tissue is notoriously difficult.
Yeah, if we're dealing with frozen archival tissue from human donors, you can't just run standard ChIP-seq and expect a clean map of the chromatin.
Oh, the degradation would just be a nightmare.
It'd be useless. So what was the leap here?
That was the crucial methodological leap. They utilized this really elegant dual-pronged approach. First, they used SNADAC-seq,
which stands for?
Single-nucleus assay for transposase-accessible chromatin.
Okay, break that down for us.
So instead of just sequencing DNA blindly, you isolate the nucleus and you flood it with a hyperactive TN5 transposase enzyme.
Basically a molecular tagger.
Exactly. This enzyme naturally inserts sequencing adapters into the DNA, but, and this is the key, it physically can only access regions where the chromatin is open.
Unspooled from the histones.
Right, so it gives you this high-resolution map of all the unlocked doors in the genome.
Okay, I like that. Let's use a library analogy here.
I'm ready.
The genome is this massive library. SNADAC-seq is basically walking through and finding out which doors are unlocked.
I like it. But an unlocked door doesn't tell you if the room is actually being used.
Right.
Or if it's completely abandoned.
Right. So to see who's actually in the room, they layered on something called nano-CT tag.
Yeah, we really should pause on the elegance of this technique for archival tissue.
It's wild.
They use nanobody-tethered tagmentation to hunt down two very specific histone modifications.
Let's give everyone a quick memory trigger for these marks.
Oh, idea.
First, they targeted H3K27 axis. That's the acetylation mark.
We can think of that as the accelerator.
The accelerator, it signifies an active working enhancer or promoter, like a glowing staff pick sign in our library inviting you in.
Exactly. And then they targeted H3K27E3, the methylation mark.
Which is the mechanical brakes.
Right, the brakes. That signals silenced, repressed chromatin, heavy chains wrapped around the forbidden sections of the library.
So that overlay is where the map really comes alive.
Yeah, by adapting nano-CT tag for frozen tissue, they could see the open chromatin via ATAC, the active accelerator marks, and the repressed brake marks.
All at a single nucleus resolution.
Yes, and that allowed them to isolate the specific regulatory networks of our target cells.
The OPCs and MOLs. The ones responsible for wrapping neurons in myelin.
You got it.
So just a quick mini recap before we move on. SNADAC-seq finds the open doors. Nano-CT tag maps the active accelerator marks. H3K27 is C, and the repressed brake marks. H3K27 E3.
Perfect summary.
And OPCs and MOLs are our myelin-producing focus. So here's a question for you listening. If you could look at the library of your own cells right now, what sections of your DNA do you think would be chained up and which would be wide open?
It's a trippy thing to think about.
It really is. So, moving on. Now that we know how to read the library, what happens when we compare different libraries across the human body?
Well, the scientists looked at 3 distinct areas.
Right, let's talk scale. They took 20 donors, isolated over 100,000 nuclei from the primary motor cortex, the cerebellum, and the cervical spinal cord, and they hit them with the transposases and nanobodies to build this 3D landscape. What did they find when they mapped those neighborhoods?
When they built that landscape, they began identifying incredibly specific lineage regulators.
Yeah.
Transcription factors that dictate what a cell does.
Like master switches.
Exactly.
Yeah.
For instance, they found that FOXN2 is the definitive transcription factor governing microglia.
The immune cells of the brain.
Right. And ZBTB20 governs astrocytes. Yeah. But the master regulator for the oligodendrocyte lineage, our myelin makers, is a gene called SOX10.
SOX10?
Yeah, and the researchers identified a massive, previously uncharacterized putative enhancer region controlling this gene.
But they didn't just observe it, right? I want to dig into how they proved that enhancer actually controls SOX10.
They had to functionally manipulate it.
Using a CRISPR interference and activation model, but not standard CRISPR. They used a catalytically dead Cas9 protein, dCas9.
Right, which is so clever.
Because it doesn't shear the DNA like normal Cas9. It acts purely as a programmable GPS vehicle guided by RNA just to find that exact enhancer sequence.
That targeted delivery is brilliant. So to prove the enhancer was an accelerator, they attached the dead Cas9 to a P300 effector protein.
What does P300 do?
It acts as a histone acetyl transferase. It forcibly acetylates the local histones.
So it artificially forces the chromatin open into that H3K27C state, accelerator state.
Exactly. And when they did this, SOX10 expression skyrocketed, and the stem cells differentiated into myelinating cells.
Wow. And then to prove the reverse?
They swapped the P300 for a KRA domain.
KRAB?
Yeah. The KRB domain recruits core pressors. It forcibly applies the mechanical brakes, essentially mimicking that HVK27E3 repressed state.
And let me guess, the moment they deployed the dCas9KRB to that enhancer,
SOX10 expression collapsed completely.
Proving definitively that this specific non-coding stretch of DNA is the master volume dial for making oligodendrocytes.
Yep. They found the switch and proved how it works.
Okay, but this brings up a critical tension in the paper for me. We've mapped the regulators, we've found the SOX10 volume dial. But early on, the authors observed that glial cells look remarkably region agnostic.
They do, yeah.
Like a cortical astrocyte's chromatin looks almost identical to a cerebellar astrocyte's chromatin.
Almost indistinguishable at first glance.
Which begs a major question. If the regulatory software of these support cells is uniform across the brain, why did Kabi's team bother mapping the cervical spinal cord?
Right.
Are we just looking at the same homogeneous glia in a different zip code?
Well, the researchers suspected the mechanical and functional demands of the spinal cord might force a divergence.
Because the spine is always bending and twisting.
Exactly. And they were overwhelmingly correct. When you transitioned from the cortex to the cervical spinal cord, that uniform landscape of the OPCs fractures.
Fractures how?
A massive genomic vault that is heavily repressed and chained shut in the brain is suddenly sitting wide open in the spinal cord.
And what vault is that?
The HOX loci.
The HOX genes. The master architects of embryonic development.
The very same.
Okay, mini recap time. The study mapped 3 CNS regions, uncovered the SOX10 enhancer controlling myelin cells, and found unique transcription factor networks for different glia.
And found the HOX anomaly in the spine.
Right. So, question for you listening. How does it change your perspective of your brain to know that the support staff, the glia, have entirely different regulatory software running depending on their exact zip code in your body?
It really makes you appreciate the complexity.
It's wild. Okay, so looking closely at the spinal cord, glial cells revealed this incredible anomaly. These HOX genes.
Yeah, so during embryogenesis, the HOX gene clusters dictate the entire anterior to posterior body plan.
Head to tail.
Exactly. They tell the dividing cells where to build the brainstem, where to drop the cervical vertebrae, where to grow the limbs.
But once you reach adulthood, that job is done. The house is built.
Right, so in the cortical glia they map, these HOX regions were buried under H3K27E3 marks.
The mechanical brakes were fully engaged, chains on the library doors.
Fully chained, but in the adult spinal cord OPCs and MOLs, the brakes are off.
Wait, really?
Yeah, but it isn't just a random unlocking. What's fascinating here is there's a highly specific physical topography to it. They found a stark sequential gradient across the HOXA and HOXD gene clusters.
A gradient? Like it changes as you move down the DNA.
Exactly. At the 3' end of the cluster-covering genes like HOXA1 through HOXA7, the ATAC accessibility is through the roof.
The doors are wide open.
Wide open. And it is glowing with those HOK27 accelerator marks.
Okay.
But as you move down the DNA strand to the 5' end, genes like HOXA10 through HOXA13, the chromatin abruptly shifts.
Shifts to what?
Into the heavily repressed H3K27E3 state.
The brakes. So open at one end, chain shut at the other.
Yes, that 3' to 5' gradient is the ghost in the machine. That specific epigenetic signature is the exact chromatin conformation of a developing fetal spinal cord.
Wow.
The physical architecture of the DNA in these fully mature adult spinal cord cells is frozen in time. It's mimicking a developmental state from literally decades ago.
But here's where it gets really interesting to me. I'm looking at this decoupling phenomenon, and it is arguably the most fascinating mechanism in the entire study.
It is so counterintuitive.
Right, because the chromatin says accelerate at the 3' end, the doors are open, the active marks are there, but when they cross-reference this with RNA sequencing...
There was almost zero robust gene expression.
None. The cells aren't actively producing these embryonic HOX proteins?
Which breaks the usual rules. Usually an active enhancer and an open promoter equal active transcription.
Exactly. It reminds me of a getaway car parked in an alley.
Oh, I like this.
The car isn't moving. There's no forward motion, which is our gene transcription, but the engine is roaring, it's in gear, and the driver's foot is hovering over the pedal.
The genome is primed.
It's decoupled from expression, meaning the potential energy is there, ready to be unleashed the absolute millisecond a signal is received.
And maintaining that primed state keeping, the engine of a getaway car running for 60, 70, 80 years of a human lifespan, that requires immense metabolic energy.
Because the DNA naturally wants to close up, right?
Yeah. Yes. The epigenetic machinery has to constantly fight off the natural tendency of unused DNA to condense and methylate. Biology does not waste that kind of energy without an evolutionary mandate.
Okay, let's consolidate this before we look at how this system gets hijacked.
Good idea.
HOX genes build the body plan. Adult spinal cord OPCs keep these fetal genes primed in a 3' to 5' gradient. The genes are open but currently silent. The engine is running.
Spot on.
So, listeners, think about a physical or mental skill you haven't used since childhood. Do you feel like your nervous system still has that engine running in the background, just waiting for the signal to act?
That's a great way to internalize it.
Right. But it brings us to the big question. If the cell doesn't need to build a spine anymore, why does it spend energy keeping the engine running on these developmental genes?
And the answer to that reveals a pretty terrifying biological trade-off.
The double-edged sword. So let's talk about the light side first. Regeneration. Are we certain this is a functional evolutionary adaptation, or could it just be like a developmental oversight?
Like the cell just forgot to clean up its workspace.
Yeah, exactly.
Cobby's team anticipated that exact skepticism. But if we connect this to the bigger picture, the physiological context of the spinal cord provides the answer.
Because it's a volatile environment.
Highly volatile. It's prone to mechanical stress, compression, inflammatory demyelination. When spinal cord myelin is stripped away, the local OPCs do not have the luxury of time.
They need to act fast.
If their HOX genes were fully repressed under layers of methylation, the heavy chains, it would take days or weeks for the epigenetic machinery to recruit demethylases, unspool the DNA, and activate the developmental pathways required to lay down new myelin.
And by that point, the exposed neuron might have already died.
Precisely. By keeping the HOX clusters primed, the OPCs bypass the chromatin remodeling phase entirely.
So the moment an injury signal is detected,
the transcription factors simply drop onto the already open DNA. The developmental cascade fires instantly, and the tissue begins repairing itself.
the primed state grants the spinal cord rapid regenerative competence.
It's an incredibly elegant survival mechanism.
But evolution always extracts a toll. And here is where we transition to the dark side of this epigenetic memory.
Yeah.
Because when you leave a getaway car running with the doors unlocked, you run the very high risk of someone else jumping in and flooring the gas.
And in this case, the carjacker is a mutation known as H3.3K27M.
Right. Let's dig into that, because what does this all mean? Are you saying the exact same evolutionary mechanism that evolved to help our spinal cords heal is the back door that cancer uses to destroy it?
Sadly, yes. This is where the biological trade-off becomes a clinical tragedy. The H3.3K27M mutation is the primary driver of high-grade pontine gliomas, or HGGs.
Some of the most devastating pediatric brain tumors we face.
What makes this mutation so insidious is its mechanism of action. A single amino acid substitution occurs in the histone itself.
Wait, let me make sure I have this right. A methionine replaces a lysine at position 27.
Yes, and because that lysine 27 is the exact target for our mechanical brakes, me3-methylation, the entire system collapses.
Oh.
The mutation functionally poisons the polycomb repressive complex, globally erasing those H3K27E3 marks across the cell.
It wipes out the brakes everywhere.
Now apply that global loss of brakes to our spinal cord OPCs. In a normal cortical cell, erasing the brakes might cause chaos, but the HOX doors are still heavily spooled and closed.
But in the spinal cord OPCs, the 3' end of the HOX cluster is already unstoled.
Exactly. It's already primed. When the H3.3K27M mutation hits an OPC with an already running engine, the aberrant activation of the HOXA and HOXD clusters is explosive.
The mutation doesn't have to pry the doors open. It just pushes the gas pedal on a system that evolution deliberately left idling.
Right. It hijacks the regenerative competence of the cell, forcing it to violently proliferate as a malignant tumor.
The very epigenetic memory that evolved to save the spinal cord from trauma is the exact vulnerability that predisposes it to gliomogenesis.
And to definitively prove this structural vulnerability, the researchers deployed micro-C, which is arguably one of the most advanced techniques in this entire paper.
Yeah, let's talk about micro-C. We are moving beyond just identifying what parts of the DNA strand are open or closed here, right?
Right. We're moving into mapping the physical three-dimensional topology of the genome inside the nucleus.
Let me break down how micro-C visualizes this for everyone, because it's essentially nucleosome resolution contact mapping.
Mm-hmm.
The researchers use a chemical cross-linker to literally freeze the DNA in place, gluing together any strands that are physically touching each other in 3D space, even if they are millions of base pairs apart linearly on the strand.
It captures the loops and folds.
Right. Then they digest the DNA, ligate those touching pieces together, and sequence them. It builds this incredible heat map of the genome's physical folding architecture.
And when they applied micro-C to induced pluripotent stem cell-derived human oligodendrocytes, the results were staggering.
What did they see?
The physical 3D looping structures of the HOX loci in these healthy adult-like cells perfectly mirrored the pathological 3D architecture found in those highly lethal H3.3K27M mutant gliomas.
So the healthy cell and the cancer cell are utilizing the exact same three-dimensional origami at the HOX loci.
The topology is identical.
The only difference is that the tumor has lost its regulatory brakes.
Yes. It forces a complete paradigm shift in how we view oncogenesis in the central nervous system.
How so?
Well, we traditionally view disease as a breakdown of normal function or an invasion by foreign elements. But here, the vulnerability is not a flaw in the system. It is a fundamental feature of the system's design.
Wow.
The capacity for rapid healing and the capacity for catastrophic tumor growth are locked together in the same spatial architecture of the DNA.
That dual nature of the epigenome is just, well, it's a lot to process.
It's profound.
So let's recap this final section. Epigenetic memory allows for rapid myelin regeneration, but this prime state acts as a vulnerability that mutant gliomas exploit. And that 3D micro-C mapping proves the physical structures match perfectly.
Exactly right.
So to our listeners, how does understanding this biological trade-off change how you view disease? Not as an alien invader, but as your body's own healing mechanisms being hijacked.
That's a heavy question.
It is. We started by unpacking how techniques like ATAC-seq and nano-cut and tag let us read the active accelerators and repressive brakes on archival tissue.
Mm-hmm.
We saw how CRISPR-guided dead Cas9 functionally proved the existence of regional volume dials like the SOX10 enhancer.
Right.
And ultimately, we uncovered that our spinal glia are operating under a completely different paradigm than the rest of the brain, preserving the ghosts of their fetal development in a primed, 3' to 5' gradient, just waiting for the signal to rebuild.
And this raises an important question, something really profound regarding the rest of our anatomy.
What's that?
If the human spinal cord actively preserves its embryonic architecture at the risk of fatal gliomegenesis, just to ensure rapid remyelination, what other latent evolutionary software programs are currently idling in the background of your liver, your heart, or your lungs?
It's just sitting there.
Yeah, what other dormant ghosts of development are sitting there, primed and waiting for the right environmental trigger?
That is unsettling, but fascinating. It completely redefines what it means for a cell to be mature. The blueprints of your embryonic past aren't discarded. They are actively shaping the vulnerabilities of your future.
Could not have said it better.
We want to thank you for joining us on this deep dive into the hidden epigenetic architecture of the human nervous system. Keep questioning the incredible machinery inside you, because the deeper we look, the more we realize the past is never really gone. Until next time.